Ever wondered how the smallest units of life function and interact with one another? Welcome to the intricate realm of molecular biology, a field dedicated to understanding life at its most fundamental level. This science delves deep into the cell, the basic building block of life, exploring its structure and the intricate workings that keep us alive and thriving. Molecular biology is like the bridge connecting several scientific disciplines, combining elements of genetics, biochemistry, and cell biology. It's a magnifying glass that lets us zoom in on the very fabric of life, the chemical composition of living organisms, and how these elements interact to create the dance of life. Central to this dance is DNA, the blueprint of life, controlling the activities of the cell. This blueprint isn't just a static set of instructions, it's a dynamic, interactive entity that responds to its environment and helps shape it. The world of molecular biology is as complex as it is fascinating. Buckle up as we unravel its mysteries. DNA, the blueprint of life, but how does it command our cells? That's a question that piques the curiosity of many. To understand this, let's dive into the fascinating world of deoxyribonucleic acid, or as we commonly know it, DNA. DNA, a molecule that carries most of the genetic instructions used in the growth, development, functioning, and reproduction of all known living organisms, is composed of two strands coiled around each other to form a double helix. Each strand is made up of a sugar phosphate backbone and attached bases, namely adenine, thymine, guanine, and cytosine. These bases pair up in a specific manner, adenine with thymine and guanine with cytosine, to form the rungs of the DNA ladder. Now, the DNA doesn't just sit there doing nothing. Oh no, it's quite the busy molecule. It's the master librarian of your cellular library, holding the code that guides the functions of the cell. This code, written in the language of the bases, determines everything from your eye color to your susceptibility to certain diseases. But that's not all. DNA also plays a critical role in cell division and growth through a process called DNA replication. During replication, the DNA unwinds and the two strands separate. Each strand then serves as a template for creating a new complementary strand. This ensures that each new cell has an exact copy of the DNA. It's like the ultimate photocopier, ensuring that the instructions are faithfully passed on from one generation of cells to the next. In a way, DNA is like a maestro, conducting the symphony of cellular functions. It holds the sheet music for the cell's operations, guiding each instrument or protein to play its part at the right time and in the right way. It's a meticulous process, a dance of molecular interactions, all orchestrated by the masterful DNA. DNA, therefore, is the mastermind behind our cellular operations, a true maestro in the orchestra of life. From hair to hormones, proteins are everywhere. But what makes them so versatile? Let's dive into the world of proteins, these incredible macromolecules that are the building blocks of life. Proteins are synthesized from the instructions encoded in our DNA, the blueprint of life. This process of protein synthesis, also known as translation, takes place in the ribosomes of our cells. Here, the genetic code from our DNA is translated into a sequence of amino acids, the building blocks of proteins. Each protein is a unique combination of these amino acids, linked together in a specific order. It's this sequence that determines the protein's structure and function. Just imagine a string of beads, where each bead represents an amino acid. Change the order of the beads, and you change the shape and functionality of the string. This is exactly how proteins work. But what do these proteins do, you might wonder? Well, their roles are as diverse as their structures. Some proteins, like keratin, provide structure and support. Keratin is found in our hair and nails, giving them their strength and resilience. Then there are proteins like hemoglobin, which carry oxygen from our lungs to the rest of our body. Without hemoglobin, we wouldn't be able to breathe or survive. Proteins also act as enzymes, speeding up chemical reactions in our bodies. For instance, digestive enzymes help us break down the food we eat into nutrients our bodies can use. And let's not forget about hormones like insulin, which are also proteins. These hormones act as messengers, carrying signals from one part of the body to another. Insulin, for example, regulates our blood sugar levels, keeping us healthy and energetic. In fact, proteins are involved in almost every process within our bodies, from fighting off infections to helping us think and remember. They are the workhorses of the cell, tirelessly carrying out the instructions encoded in our DNA. So whether it's the curl in your hair, the beat of your heart, or the thought in your mind, it's all thanks to proteins. Proteins, the workhorses of the cell, are truly the embodiment of DNA's instructions. What links DNA to proteins? Enter the central dogma of molecular biology. The central dogma is the core concept that illuminates how life works at a molecular level, serving as the blueprint of life. It describes the flow of genetic information within a biological system. Its path is straightforward and unidirectional, from DNA to RNA and finally to proteins. Let's break it down further. We start with DNA, the carrier of our genetic information. It's like a cookbook full of recipes, where each recipe signifies a gene that codes for a specific protein. However, the DNA itself doesn't leave the nucleus of the cell. So how do these recipes get to the kitchen, or in cellular terms, the ribosome, where proteins are made? The answer is transcription. Transcription is the process where an RNA molecule, specifically messenger RNA or mRNA, is synthesized from a DNA template. It's like copying the recipe from the cookbook. This process happens in the nucleus, where the DNA is unzipped, and only one strand, the template strand, is used to create a complementary mRNA molecule. This mRNA, carrying the copied recipe, then leaves the nucleus and travels to the ribosome, the protein-making factory of the cell. And here we come to the second part of the central dogma, translation. In translation, the mRNA is read by the ribosome, 3 nucleotides at a time, also known as a codon. Each codon corresponds to a particular amino acid, the building blocks of proteins. Transfer RNA or tRNA carries the appropriate amino acid to the ribosome, where it is added to the growing protein chain. This process continues until a stop codon is reached, signaling the end of protein synthesis. So, there you have it. The central dogma, in its simplest form, is a 2-step process: transcription, where DNA is copied into RNA, and translation, where RNA is used to build proteins. The central dogma, a fundamental principle of molecular biology, is the bridge that connects DNA to proteins. How do genes shape us, and can we shape them in return? Let's delve into the world of genes, those tiny architects of life that work behind the scenes, quietly shaping us into the beings we are. Genes are segments of DNA, each holding a specific set of instructions that determine our traits. From the color of your eyes, the texture of your hair, to your susceptibility to certain diseases, everything is written in the language of genes. Now imagine if we could rewrite this genetic language, if we could manipulate genes to produce desired traits, to cure genetic diseases, or even to create organisms with entirely new capabilities. Well, that's not science fiction. It's a field of study known as genetic engineering. Genetic engineering involves the deliberate modification of an organism's genes. Scientists use various techniques to accomplish this, such as recombinant DNA technology, where DNA molecules from different sources are combined to create new genetic material. This new material can then be inserted into an organism, giving it new traits. Consider the creation of genetically modified crops. By inserting genes that make the crops resistant to pests or tolerant to harsh weather conditions, we can increase agricultural productivity and ensure food security. Similarly, in medicine, genetic engineering is used to produce insulin, a life-saving hormone for people with diabetes. But it's not all rosy. Genetic engineering brings with it ethical and biosafety concerns. Questions about the long-term effects of genetically modified organisms on the environment and the ethics of altering human genes are hotly debated topics. Regardless of these debates, one thing is clear. Genes are not just the building blocks of life. They're the blueprint, the script, the code that makes us who we are. And genetic engineering is like having the power to edit this code, to rewrite the script of life. Genes hold the key to our identity, and genetic engineering is our tool to unlock their potential. So, how does molecular biology fit into the larger picture of life? Well, let's take a moment to recap what we've discovered today. We've learned that DNA, the genetic blueprint, is at the heart of life's processes. It's not just a static storage unit, but a dynamic, active participant in the goings-on of life. We then delved into the world of proteins, the workhorses of our bodies, carrying out the instructions written in our DNA. We saw how the central dogma of molecular biology provides a framework for understanding how information flows from DNA to RNA to proteins. And finally, we touched upon genes and genetic engineering, showcasing how our understanding of molecular biology allows us to manipulate these elements to our advantage, potentially revolutionizing medicine, agriculture, and more. Molecular biology, a complex web of interactions, is the language of life. And now, you speak a bit of it too.